everyone, and welcome to Miss Estruch Biology, and in this video I'm going through an entire topic summary of 2.11 cell structure for OCR A A-level biology. So get yourself ready, pen and paper, make some notes. We're summarising the whole thing. Now if you do want extra help with those notes, I actually have a full set of notes for OCR A biology already available just here, and I'll link it in the description below. So if you want a full set of notes that covers all of the theory for your spec, highlights the keywords, key marking points, then definitely check that out. But for now, let's get into it. So here is your summary of 2.11 cell structure for OCR A-level biology. And if you are new here, then make sure you click the subscribe button so you don't miss out on any of my latest videos. And I go on to Instagram and TikTok daily for even more advice. So the first thing you need to know is about the 4 different types of microscopes. Here's a very basic summary, but we will go into each type in a little bit more detail. So for our light or optical microscopes, they have a poor resolution, and that is because of the wavelength of light, and light is what is used to create the image. You can use living samples though, and that means that you can have a colour. You can use living samples though, and you can get a colour image. Transmission electron microscopes have a much higher magnification and resolution, and in these microscopes the electrons pass through the specimen to create an image. Scanning electron microscopes very similar, except the electrons are bouncing off the surface, so you actually create a 3D image. And then lastly, the laser scanning confocal microscopes. These are high resolution and 3D, and it uses laser light to create the image. So we'll go through all of those in a little bit more detail. But first, let's see what we mean by this term resolution and magnification, which was used in that summary. The resolution is the minimum distance between 2 objects in which you can still view them as separate. And in a light microscope or optical microscope, this is determined by the wavelength of the light. Whereas electron microscopes, it's determined by the wavelength of an electron. Magnification refers to how many times larger the image is compared to the actual object that you are viewing. So with the light microscopes or optical microscopes, there are 4 types of slide preparation that you need to be familiar with: dry mount, wet mount, squash slide, and smear slide. The dry mounts are when thin slices or even the whole organism or specimen are viewed. And what you would do is place that specimen on top of your glass slide with just a cover slip placed on top. So this might be used if you're examining a really thin slice of plant tissue, or you might have tried this before with your own hair. Wet mounts are more common, and you would have done this potentially in lower school when you looked at maybe a swab of your cheek cells or onion cells. And this is when the specimens are added to water or maybe a stain before you have the cover slip lowered on top with a mounted needle to prevent air bubbles forming. And if you were going to examine living aquatic organisms, you would have to do that as a wet mount. Now in A-level, you might be more familiar with a squash slide. So these are wet mounts, but then you would push down on the cover slip to ensure that you have a really thin layer of cells so that the light can pass through. So when you did your root tip squash practical, you would have taken a very thin slice from the tip of the root of an onion or garlic, placed the stain on top, squashed it by pressing down, and that way you should get light passing through so you could visualize and see the chromosomes. And then finally, we have the smear slides. These are created by placing a drop of the sample at one end of the slide and then using the edge of another slide held at an angle to smear the sample across that initial slide. A cover slip is then placed on top. And this is used when examining blood cells in a blood sample, for example. Next, we move on to this math skill, the eyepiece graticule, and calibrating it. So inside a light microscope, in the eyepiece, there is actually a scale that you can insert on a glass disc, and that is what the eyepiece graticule is. And the point of it is that when you look through the microscope, you can use that scale to measure the size of the object you're looking at. But there are different lenses on your microscope which are going to be causing different magnifications. So you might have times 10, times 40, times 100. And at each magnification, the value of one of the divisions on your eyepiece graticule will be different. So you have to know how to calibrate it at every magnification to work out what one division on your eyepiece graticule is worth. So this is how you would do that. You would need to use something called a stage micrometer. And this is a glass slide which essentially has a ruler on it. And you would place that on the stage of your microscope, look through your eyepiece, and then move your stage micrometer so that it is aligned right next to your eyepiece graticule. So on this image, the circle's representing your field of view when you're looking down the microscope. The bottom scale is the scale on your stage micrometer, and that has been aligned against the eyepiece graticule, which is already within the eyepiece of the microscope. Once you've then got them aligned, step 2 is to count how many divisions on the eyepiece graticule fit into one of the divisions on the stage micrometer. In this case, we can see 2 divisions fit into one of the stage micrometers. Now, on a stage micrometer, each division is worth 10 micrometers. That's why that value there says 10. It's one division, but that one division is worth 10 micrometers. So we can then use that to work out at this magnification what is one division worth on the eyepiece graticule. So if we know that one of these divisions is worth 10 and 2 divisions fit into one of those, is 10 divided by 2. So we know that at this magnification, one of these divisions on the eyepiece graticule is worth 5 micrometers. So you can then measure your specimen to work out what is the actual size of your specimen. Something else that you can do in terms of calculations with the light microscopes, but even with electron microscopes as well, is the magnification calculation. So you need to know that to work out the magnification, it's the size of the image divided by the size of the real object. And you might have to rearrange that formula to work out one of the other components of this equation. And it's never usually that straightforward. Often you have to have changing of units involved as well. So we usually measure the size of an image in millimeters, and the size of the real object is usually in micrometers. So to convert your millimeters into micrometers, you would have to multiply by 1,000. So there's normally that additional step as well. So staining is the next thing you need to know about. And some cell components are really difficult to see under the microscope unless you add a stain to make it a much more obvious colour so it stands out. And differential staining is a technique which involves many chemical stains being used to stain different parts of the cells different colours. Again, just to make it visually more obvious what you're looking at. So crystal violet or methylene blue are 2 stains that are commonly used, and they are positively charged stains. And that means they'll be attracted to anything that is negative. So negatively charged components of the cell, and they will stain those parts of the cell. You could then also add nigrosin and Congo red, which are negatively charged, and therefore they actually can't enter the cells because the cytosol would repel it. And that would create a stained background, a different colour, and the unstained cells will then stand out. Observations of structures from under a microscope can be recorded in the form of scientific drawings, and there are set rules that you need to know for how you create a scientific drawing, because these are very different to your typical artistic shaded style drawings. So number 1, it should always be in a pencil. Title the diagram to indicate what the specimen is. You have to state what magnification was used for the microscope and that you have drawn the image at. You should be labelling the key features as well. So for a high power plan of this would be limited to just the nucleus, cytoplasm and plasma membrane. You also have to annotate cell components, cells and sections of tissues that are visible also, and stating what colours and shapes they are, so not necessarily their functions. So then point 6 is the bit where we're saying it's not like an artistic diagram, so you don't do sketches. So you should use solid lines that touch, but there are no overlaps between those lines. Also, there shouldn't be any colouring or shading in. And then lastly, the label lines should be horizontal, drawn with a pencil and ruler, and not have arrowheads on them. So essentially, the aim of a scientific drawing is to show the size,
location and proportion, and annotating some of these structural details.
You could also be asked to do the scientific skill of scientific drawings, and these are very different to artistic drawings. They have a complete set of rules, and it includes when you are looking at your specimen under the microscope, you would then try and draw it using, first of all, it has to be a pencil, a really sharp pencil so you can be accurate. You have to include a title of what you've drawn. You have to state the magnification, or you could include a scale. You should always annotate the cell components or the cells themselves or the section of tissue that is visible that you've drawn. It shouldn't be sketchy lines. It has to just be solid lines that don't overlap with no gaps, and you mustn't do any colouring or shading. So essentially the aim of these diagrams is to show the size, location, proportion. They are not artistic, so that's why we don't have any sketching, shading or colouring. It's all about the facts, the size, shape, position and labelling the structures. And although I've said this for microscopes, when you come on to do dissections, you could be asked to do scientific drawings of what you observe in your dissection as well. So then we move on to the electron microscopes. And a beam of electrons is used here to create the image. And electrons have a very short wavelength. And that is why electron microscopes have a higher resolution. And because they've got a high resolution, small organelles and internal structures can be visualized. The image is created using an electromagnet. So that magnet is used to focus the beam of electrons. Now, you can't actually use living samples with electron microscopes, and that's because the air would absorb the beam of electrons. So your electron microscopes have to be used with a vacuum. So your specimen has to be in a vacuum so that the air doesn't absorb the electrons. The image is also in black and white, so you do have to add a stain to add any colour, or you can do that artificially afterwards. So transmission electron microscopes are one type of electron microscope. And for these ones, your specimen has to be very, very thin. You're using an extremely thin slice through your specimen. They're stained and then placed in a vacuum. An electron gun will then be used to release a beam of electrons. The electromagnet will focus that beam, and these transmit or pass through the specimen. Some parts of the specimen will absorb the electrons, and that will make them look darker in the image, and some parts won't and they look lighter. So you get a 2D image because the electrons are passing through. And these are really useful for being able to see the internal structures of cells. Scanning electron microscopes, in these ones, the specimen doesn't have to be thin because the electrons don't pass through the specimen. Instead, the electrons are beamed onto the surface, and the electrons scatter in different ways, so they reflect backwards in different ways depending on the contours of your specimen.
And this is what produces a 3D image of the surface of the specimen.
The laser scanning confocal microscope, this is a type of fluorescent microscope, and the image is created using a very high light intensity to illuminate the specimen stained with a fluorescent dye. So this combines the benefits of high resolution optical imaging with depth selectivity, which we can see in this diagram or image. It's slightly out of focus, but you can see that you do have that 3D depth to the image. So this enables scientists to view sections of tiny structures that would be challenging to physically section off, such as in an embryo, to create a 3D image. And the image is created as the microscope scans the specimen point by point using a focused laser beam to create a 2D image or a 3D image in different focal planes. As the light is emitted from the specimen, it creates fluorescence. So the next bit of spec then is knowing the organelles you find in eukaryotic and prokaryotic cells. You need to know the structure and function. And here are the 13 organelles that you need to know for eukaryotic cells. And eukaryotic cells include animals, plants, and fungi. So here's a cross-section of an animal cell showing some of those structures. And here's a cross-section of the plant cell, so you can see the location and general shape of these organelles. But we're going to go through each of them in detail, starting with the nucleus.
The key structures are that you have a nuclear envelope, which is this double membrane layer, and the holes that we can see within it are the nuclear pores, and that is what the mRNA can pass out of after transcription. There's also nuclear plasm, which is a granular jelly-like material in the middle. The chromosomes are found inside of the nucleus, and these are protein bound, so wrapped around histone proteins, and they are linear. Lastly, we have the nucleolus, which is this smaller sphere inside of the nucleus, which is the site of RNA production, and it makes ribosomes. So the function then is, first of all, it's the site of DNA replication and transcription. And transcription is the first stage of protein synthesis when mRNA is created. It also contains the DNA for each cell, and it's the site of ribosome synthesis.
Flagella are not actually found on all eukaryotic cells. They're found on some, so for example, sperm cells. And it's this whip-like tail structure, and the function is for mobility. It is also sometimes as a sensory organelle for chemical stimuli. Cilia, again, are not on all cells, and these are hair-like projections that come out of the cell itself. And they might be stationary cilia, or they might be mobile. And mobile cilia move to help sweep substances along. And that could be in the trachea, for example. They'll be moving in this wave-like motion to help sweep the mucus up and out of your trachea to prevent lung infection. Stationary cilia are important in sensory organs, such as your nose. Centrioles, these are made up of microtubules, which we can see here in this image, the microtubule. And they occur in pairs to form a centrosome. So that's what we're looking at here. We've got them as a pair forming that centrosome. Now these are involved in the spindle fibre formation, which is essential in organising the position of chromosomes in mitosis and meiosis. The cytoskeleton is a network of fibres found within the cytoplasm all over a cell. And it consists of microfilaments, microtubules, and some intermediate fibres as well. Now, the function of this is it provides mechanical strength to cells, it helps to maintain the shape and stability of a cell, and many organelles are actually bound to that cytoskeleton to hold it in a fixed place. Microfilaments are responsible for cell movement, and microtubules are responsible for creating scaffold-like structures. The intermediate fibres help to provide mechanical strength.
We then move on to looking at the endoplasmic reticulum. In terms of the structures, there's two types. We have rough and then we have smooth endoplasmic reticulum. Both of them have folded membranes, which are called cisternae, but the key difference is the rough have ribosomes attached to the outside, whereas the smooth doesn't. The difference in function then links to the fact that the rough has the ribosomes, because the rough endoplasmic reticulum has the function of protein synthesis for proteins destined to leave the cell, and the proteins are transported through the rough endoplasmic reticulum and into secretory vesicles. The smooth endoplasmic reticulum is where lipids and carbohydrates are synthesized and stored. Next then we look at the Golgi apparatus and the secretory vesicles. This is the Golgi apparatus and the secretory vesicles here, which I've seen many students describe as looking a bit like a Wi-Fi symbol, and that's how they recognise it in comparison to the smooth endoplasmic reticulum. But what it is folded membranes making the cisternae. You then have secretory vesicles that pinch off from that cisternae. The function then is, it's where we have the addition of carbohydrates to proteins to form glycoproteins. You also have the production of secretory enzymes. Carbohydrates are secreted. We have the transport, modification and store of lipids. We get lysosomes forming. Molecules are labelled with their destination. And then finally, the finished products are transported to the cell surface membrane, which we can see here, in those secretory vesicles where they fuse with the membrane and the contents are released.
Lysosomes. So we just said lysosomes can be created by these Golgi apparatus, and lysosomes are vesicles and they're bags or vesicles of digestive enzymes. And they can contain lots of different enzymes. Some functions include the hydrolysis of bacteria. So you have lysosomes fusing with phagosomes in phagocytosis. That releases the digestive enzyme to hydrolyze and destroy pathogens. They're also involved in completely breaking down dead cells. And then once they have hydrolyzed and digested whatever it is that they're breaking down, the lysosome will then fuse with the cell membrane, and that will release its contents to the outside of that cell. Mitochondria are double membrane-bound organelles. So we have an outer membrane, and the inner membrane is shown here in yellow. It folds in to create the cristae. The mitochondria is the site of the fluid center in the middle is called the mitochondrial matrix, which is the site of some of the stages of aerobic respiration. And they actually also contain their own ribosomes and loops of DNA so that they can create the enzymes necessary for respiration inside of the organelle itself. So it's the site of aerobic respiration specifically. Therefore, it's the site of ATP production. And as I've already said, it contains the DNA to code for the enzymes needed for respiration. So the ribosomes it contains are the 70S ribosomes, which are much smaller, which are also the type that are found in prokaryotic cells. So that leads us into ribosomes. Ribosomes are small. They're very, very small. You can see they're just like these tiny dots in the microscope images. They're made up of 2 subunits of protein and RNA. So those are the 2 molecules it contains. There are ATS ribosomes, which are the larger ribosomes found in the cytoplasm of eukaryotic cells, or 70S ribosomes, which are much smaller, found in prokaryotic cells, and also in mitochondria and chloroplasts for eukaryotes. And the function is, it's where protein synthesis occurs. Chloroplasts are found in plant cells, which are eukaryotic organisms. They also have a double membrane, and then inside you have these foldings of even more membrane, which are called the thylakoids. And these foldings stack up to look a bit like coins, and we call those stacks the grana for plural or granum for singular. And you also have this fluid, which is shown in this beigey orange colour, which contains lots of enzymes needed for photosynthesis. So this is the site of photosynthesis. Cell walls are found in plants and fungi of eukaryotic organisms, not in animal cells. And in plants, you have microfibrils of the cellulose polymer. And fungi, you have chitin instead. So they're made up of 2 different molecules. Plants have cellulose for structural strength, whereas fungi have chitin. And that is still a polysaccharide, but it also contains nitrogen. The function of the cell wall, though, is to provide structural strength. The plasma membrane is found in all cells, and this would form the cell surface membrane. It's made up of this phospholipid bilayer, and within that bilayer you have other molecules embedded, such as channel proteins, carrier proteins, proteins on the outside which might act as receptors, glycoproteins which might act as receptors. You have cholesterol, which affects the fluidity and therefore the permeability of the membrane as well. And all of these structures together help to control what can enter and exit the cell. Or it could be the organelle, because you do have these membranes on the outside of some organelles as well. So some of those organelles are involved in the production of proteins. And you do get long answer questions where it's in this topic, but they want you to link together everything you've learned to say which organelles are involved in the production and secretion of proteins. So let's summarise that. Number 1, the polypeptide chains are synthesized on the rough endoplasmic reticulum, or you could say the ribosomes, because those are found on the outside. Those polypeptide chains move to the cysteine in the RER, and they get packaged and they get folded and packaged into vesicles to be sent to the Golgi apparatus for further modification. Now they have to move through the cytoskeleton to get to that Golgi apparatus. Once they're inside the Golgi apparatus, the proteins are modified further and packaged into the vesicles, and those secretory vesicles carry the proteins to the cell surface membrane. The vesicle then fuses and releases the protein by exocytosis.
So looking at the prokaryotic cells then in comparison to the eukaryotic cells, here are the key differences. First of all, prokaryotic cells are much smaller in size. They don't contain any membrane-bound organelles. Both have ribosomes, but prokaryotic cells have the smaller 70S ribosomes, whereas eukaryotic cells have the larger ATS ribosomes. The DNA in a prokaryotic cell is not contained within the nucleus, And prokaryotic cells do have cell walls, but they're made up of peptidoglycan, whereas in eukaryotic cells, in plants it'd be cellulose, and in fungi it'd be chitin. Now, some prokaryotic cells have additional features, which are the plasmids, a capsule around the cell, and the flagella.
So let's have a look at some of those key differences. So we said there's no membrane-bound organelles. So that means they don't have their circular DNA in a nucleus. They don't have mitochondria, chloroplast, a Golgi apparatus, and endoplasmic reticulum. They're much more basic organisms. The ribosomes, we said they do still have ribosomes, but they're much smaller. So they are the 70S ribosomes compared to the 80S ribosomes found in eukaryotic cells. They don't have a nucleus, so instead of a nucleus, they have their single circular DNA just free within the cytoplasm, and it is not attached to proteins. And that is what this image here is showing. Now, that is not the same thing as plasmids. Plasmids are additional loops of DNA that some bacteria have. They don't all have them, and they only have a few genes on them, and it's where you'd find the genes for antibiotic resistance. And bacteria either don't have them at all, or they might have them, but they have them in varying numbers.
The cell wall of a prokaryotic cell is made up of peptidoglycan, which is a glycoprotein. In comparison, eukaryotic cells, animals don't have a cell wall, but plants and fungi, which are eukaryotes, do. And plants have a cell wall that's made up of microfibrils of the cellulose polymer, and fungi have a cell wall made up of chitin, which is a nitrogen-containing polysaccharide.
The capsule, which again only some bacteria would have, this is a slimy layer made of protein on the very outside. And the function is to prevent the bacteria from drying out or desiccating. But it also helps to cover the antigens to make it harder for the host's immune system to detect the bacteria. The flagellum, which some bacteria have, and some might have multiple, some might only have one, some won't have any. Just like a flagella in a eukaryotic cell, it rotates, and its function is to move the bacterium. So that's it. That covers all of cell structure for topic 2.11 for OCR A. Make sure you are subscribed so you don't miss out on any of these summary topic videos. And if you do want more helpful free to do with exam technique, then check out my free guide in the description.